This short video aims to highlight the molecular cause of PKU using the mutation known as R408W as an example. As the complete structure of the phenylalanine hydroxylase enzyme is not currently available, only two of the enzyme protein subunits are shown here. Each individual subunit has three protein domains, a catalytic domain, shown here in blue and pink. This is the active component of the enzyme. There's also a tetramerization domain or oligomerization domain, shown here in yellow and in orange. And finally, there's also a regulatory domain, but this is not depicted here. Firstly, in the case of the non-R408W mutant, if we zoom in on the structure and orientate ourselves, we can see that the residue at position 408 is the amino acid arginine. This forms links with other amino acids part of the structure within the same subunit. These links are called hydrogen bonds, and we can see three of them here. In this case, what these hydrogen bonds links do is that effectively they hold the catalytic domain in blue against the oligomerization domain in yellow. They do this in a specific manner which orientates the oligomerization domain of this subunit against the oligomerization domain of another neighboring subunit, shown here in orange, and this promotes formation of the tetrameric structure. However, in the case of the occurrence of the R408W mutation, there is a single amino acid substitution at position 408, which sees the replacement of arginine with another amino acid known as tryptophan. Importantly, you can now see loss of those hydrogen bonds, shown before. This means that the oligomerization domain loses its specific orientation conferred by those bonds. And this has been shown to result in protein aggregation and complete loss of the tetrameric form of the enzyme. Effectively, these protein aggregates are non-functional, and therefore you get increased levels of phenylalanine in the blood, which leads to the symptoms of PKU.